Welcome to Existential Medicine, a new lab in Johnson & Johnson J-Labs series with supporting partner EY. I'm Alexandra Bacchus and I'm part of the new lab team. Many of you might be familiar with our work here, but others might be encountering our work for the first time today. Thanks for joining us. For those of you not familiar with New Lab, we're a community of experts and innovators applying transformative technology to solve the world's biggest challenges. Through our membership and studios, we bring together entrepreneurs, engineers, inventors, and industry leaders to create sustainable solutions and enterprises. And we employ technologies including robotics, AI, and material science to transform what matters most: health, environment, media, cities, and infrastructure. That mission has taken on a whole new meaning during the pandemic. Today, we'll be discussing how imaging technology has evolved and altered the way we view the brain and neurodegenerative diseases. This installment of Existential Medicine will explore how transformative technology has reshaped scientific beliefs, informed basic research, and driven patient care decisions. A few housekeeping notes before we kick things off with Orlin. Throughout this hour, we're going to conduct interactive polls, so please keep an eye out for those questions popping up and please do weigh in. We love to see your perspective reflected in the results. Additionally, our panelists are looking to field questions from the audience. To ask a question, please type it out in the Q&A button and I will get those questions in front of our panelists. So without further ado, I'd like to pass it over to Orlin Boston, EY Global Accounts Excellence and Innovation Leader.
Thanks, Alexandra, and exciting that here we are on our 8th installment of Existential Medicine. Going back 2 years ago when New Lab and J-Labs and J&J Innovation, along with EY, came together to sort of think about what could we produce that could be interesting about the future of medicine. And here we are 8 installments in. For those of you who joined us for our 7th installment, which was focused on patient data, privacy and security, we really delved into some issues around challenges, some of the provocative issues that arise as we sort of push the envelope in that area. We'll be doing the same thing in this next session, focused on seeing the brain, neuroimaging, and neurodegenerative diseases, and really the impact of these imaging technologies. Because look, I think as we think about the future of neurodegenerative diseases and treatments for those are going to highly depend on our ability to advance neuroimaging technology. So looking forward to this discussion, and again, excited to be partnering with J&J, J-Labs, and New Lab. With that, I'll pass that on to Stephen.
Yes, Stephen, please feel free to turn on your video. He is the head of J-Labs New York, which is incubating 100+ healthcare startups across Johnson & Johnson's healthcare divisions.
Yeah, thanks. Hi, everyone, and thanks for joining us this afternoon. It's great to be here. I think as Alexandria mentioned, my name's Stephen Pitt. I am the regional head of J-Labs across the Northeast here. As you may know, J-Labs is a no strings attached incubator arm of Johnson & Johnson Innovation. Our New York space is based right out of Soho, and we're always accepting applications on an ongoing basis from startups across all areas of life science. That includes pharmaceuticals, consumer medical devices, and health tech. J-Labs, to echo Orlin's comments, I think J-Labs is very proud to be one of the founding co-founders of Existential Medicine, along with New Labs and EY. This is our 8th installment, as Orlin said as well. I think it's been super interesting, the 1 or 2 that I've been to so far. Really proud to be associated with this. I think there's all the topics that proved to be very provocative, cutting edge. I really like the last one I went to on basically on data privacy. I think it's so apropos right now with the current environment that we're in. So really thrilled to be on the cutting edge of all these great different topics that we're going through. So at J-Labs, neuroscience is one of our key focus areas, and will continue to be through at least next year. So really thrilled to be here, really excited for today's discussion on computational neuroscience and imaging. And thanks again for everyone for joining us, and we'll have a great session. Thanks.
Thanks so much. And lastly, but of course not least, I'll pass it over to Dan Seawald, founder and CEO at Deliberate Innovation.
All right. Well, thank you, Alexandra, and also big thanks to Stephen and Orlin. And in a moment, I'm going to introduce our panelists, but one quick say that I like to have is this is a topic that's near and dear to my heart. Having done work in neuroscience and really care a lot about neurodegenerative diseases and what we can do in the future, imaging may very well be at the heart of this. So we're going to start broad and then we're going to narrow, and we're going to meet three of the leading minds in this space and find out more about what's happening, but also taking a step back and just understanding some of the fundamentals, things I think will be very intriguing. So let me invite Paul, Mackenzie, and Hartmuth to turn their cameras on and join me on the screen. I'm going to take a moment to, first of all, thank you guys for joining. And I'm going to take a couple more moments to tell you why I think they're the leading minds in this space. I'm going to start by first introducing Dr. Paul Mccracken. Paul is the Vice President, Global Head of Medical Imaging at the North Wales Site Head at ICON PLC. Now, you may be wondering, who is ICON? Well, ICON is a global provider of outsourced development and commercialization services to the pharma, biotech, and med device industries. Paul's been working for over 20 years in imaging and pharmaceuticals, so he's not a newcomer by any stretch, and he's got a strong record of applying imaging and biomarkers in drug discovery and development across many different therapeutic areas. Before Paul came to ICON to do the work that he's doing there, he founded and led an imaging center of excellence at Eisai, a Japanese pharmaceutical, and has previously worked at the Merck Research Labs developing preclinical and translational imaging biomarkers and strategies to assess new molecular and biologic entities. Oh, and by the way, Paul earned his doctoral degree in biomedical engineering, imaging from the Mayo Clinic, and Paul regularly presents on the use of imaging and biomarkers. So we're looking forward to hearing from you, Paul, and thank you for joining.
Thank you, Dan.
Mackenzie. Dr. Mackenzie Mathis is an assistant professor at the EPFL and the Bertarelli Foundation Chair of Integrative Neuroscience in Geneva, Switzerland. So thank you for staying up later in the afternoon, evening, Mackenzie. The EPFL, they're a part of the Brain Mind Institute, or the Bertarelli Integrative Neuroscience is part of the Brain Mind Institute at EPFL, and the Center for Neuroprosthetics and the Center for Intelligent Systems. Lab studies adaptive motor behavior in mice, performs large-scale recordings of neuropopulations. They build new robotic tools for neurocircuit interrogation and behavioral studies, as well as machine learning tools for behavioral analysis. So quite a lot. We're going to delve into that. Before that, Mackenzie was a faculty member at Harvard, and she completed her doctoral studies at Harvard University. So thank you for joining, Mackenzie. We'll be hearing from you in just a moment.
Thanks, Dan, for having me.
And last but certainly not least, Dr. Hartmuth Cole. He received his doctoral degree in organic chemistry in 1991 in London at the Imperial College. Following, he did postdoc work and he joined Ciba-Geigy in 1993. And for a period of about 5 or 6 years, Dr. Kolb developed the click chemistry approach. And in 2002, he worked at Scripps Research Institute as an associate professor. For the next decade, he was the head of Siemens biomarker research, where he and his team developed PET tracers, and you'll be hearing more about that, and which they were recently approved by the FDA. So Dr. Kolb has now been for the past 6 years working at Janssen, and his research interests include the development of PET tracers for all therapeutic areas and the discovery of biomarker signatures for patient screening, diagnosis, and predicting treatment response or toxicity. And Dr. Kolb serves as the co-chair of the Neuroscience Steering Committee of the Foundation of NIH Biomarkers Consortium. Hartmuth, thank you for joining. Great to have you.
Thanks for inviting me. Thank you.
All right. Well, that was a mouthful. Now we know your professional CVs. I want to know just a little bit more about what you guys do during the day. So, Paul, if you don't mind, I'm going to come back to you. You have an amazing biography, but can you tell me a little bit about what do you do during the day and how it connects to the topic that we're here for?
Yeah, thanks, Dan. And thanks again for inviting me to participate today. So as you mentioned, ICON, it's a global clinical research organization. So from large pharma to biotech with a single asset, our role is to help those organizations partner and develop those assets and really interrogate whether or not they're bringing benefit to human disease as intended. As the head of the medical imaging department, whenever there's a trial that has an imaging component, and for neuroscience in particular, where imaging is oftentimes the only way to understand what's going on within the brain. We would participate in any aspect of that trial from phase 1 through phase 4 and really work with the sites, work with the sponsors, help them develop a plan and talk about what it is they need to do to develop their asset and really interrogate it. And I'll speak a bit more about what that means later on in the conversation.
And I'm just curious, Paul, how standard has imaging been part of trials? Has that been a long-standing tradition, or is that becoming more and more commonplace in the past 5 or 10 years? What's the receptivity like of imaging being a standard part of even later trials?
Yeah, without question, imaging is part and parcel to the vast majority of clinical trials that move forward. A good example is oncology. I think there's probably very few exceptions with the many oncology trials going on right now where imaging isn't a significant contributor. But I'd say in other areas such as NASH, musculoskeletal disease, and particularly, again, neuroscience, imaging has, especially as we've developed new tools, and again, I think we'll speak about those in a bit, you're seeing an increased uptake in the ability to use those not only in phase 1, 2, but as well as phase 3 to help really drive decisions and make sure that the compounds are going to where they're supposed to be going, and that the mechanism of action, the drug is acting as how you'd expect, and then even contributing some part to efficacy as well.
Great, thanks. We'll go much deeper. Thanks for sharing about what you do during the day. Mackenzie, I suspect you do something a little different during the day. I'd love to hear more about what do you do that goes beyond the bio that we shared? What is your everyday like, so to speak?
Sure. Yeah, so as you mentioned, I'm situated in Geneva now at the Center for Neuroprosthetics. And so that means I sit, one with my lab team. I'm an assistant professor, which means I spend still a lot of time doing experiments myself. And so we generally have mice as our model organism, which I'm sure we'll dive into much more later today. But in general, we're really interested in how the brain is adapting and how it changes. And we really use this as a proxy for also changes later in neurodegenerative diseases. And so we want to be able to understand how animals can easily adapt to their environment. And so it might sound quite funny to say, but we essentially teach mice to play video games, and then we study their brain and how they change during these different epochs of the game. And so we want to understand how these algorithms in the human and, well, eventually the human brain, but in the mouse brain could be translated to potential human therapies later. So really our day can span from anything from taking care of mice to doing surgeries to implanting little glass windows so we can image the brain, to doing computer simulations, and then having a lot of team meetings. And now, part of COVID, of course, a lot of Zoom webinars.
Who doesn't love a good Zoom webinar? Maybe the mice don't.
Yeah, I haven't had any mice on Zoom, so.
That'll be the next one. Before we move on to Hartmuth, one quick question. Why mice? That was one of the things that crossed my mind. Why mice versus any other species?
Yeah, no, it's a great question. So in general, I mean, humans and mice, we share about 85% of the protein coding genome. So we're actually much more similar than you might think. We're mammals, and mice are actually quite skilled. They're very dexterous with their little hands. And so they're actually quite a good model organism for motor diseases and motor learning in particular. They're really agile, they're super smart, they're small and compact and have a short, breeding cycle. So all these things make them really attractive model organisms for studying. And we have an amazing toolbox that I would love to get into later to essentially record from different and really detailed cellular resolution of different cell types in the brain, which is not possible in humans or even other larger primates. Yeah.
Interesting. All right, we'll definitely get deeper on that. I also want to note that they make good pets, but I'll
They are. They're actually really social animals too, which is quite nice.
Awesome. Hartmuth, same question for you. Tell us a little bit about what, beyond the CV and the awesome work that you've been doing and have done, what do you do during the day now?
The main focus of our work is two things, really. One is to support clinical trials and allow Janssen to run better clinical trials. And I'll get to that in a second. And then the second thing is to understand the pathophysiology of disease better, right? So basically through imaging and other ways. So for the understanding of disease, we are developing new tracers that could probe certain aspects, for example, inflammation, synaptic density, and just things like that, right? And for clinical trials, there we have to be a lot more pragmatic than that, right? Because we can't, we have to be focused and be useful. And there we focus on two things again. So one is to use established imaging tracers to help inform the trial for patient enrollment, patient staging, monitoring of disease, and I'll get to the details later in the conversation, but also to develop non-invasive, less expensive tools for running trials, and there will largely be fluid markers, right? So we're using imaging to validate plasma biomarkers, which can then be surrogates, right? And at the beginning, so you can't scale a phase 3 trial just using imaging, way too expensive and too slow, right? So especially post-approval, you don't want to be dependent on imaging to get to use the therapy, right? So that's why we want to have a fluid marker as a diagnostic.
So scale and economics really does play in imaging and your weapon of choice, if you will, in the work that you do.
Yeah. I mean, as you move more and more towards approval, you need to also worry about the economics and the usefulness, right? Because if it's too expensive, then no one will use it, right? And we know the currently approved imaging biomarkers for amyloid and tau, they're not reimbursed, and the market is very small, right? And so people don't get the benefit of having these tools available because you just can't afford it, right? And so it's important to have the whole package, the therapy and matching diagnostic validated and used.
We're going to come back to that in a moment. What I want to do is take a step back for a moment. Now, without having surveyed our audience, or maybe a wide array of experiences of knowing a little bit about the brain and neuroimaging to being an expert. But I'm going to take the step back and ask the question that I would have asked if I jumped in on the panel. And Mackenzie, if you don't mind, I'm going to start with you. If we delve into the brain and imaging, how far has our understanding of the brain evolved or come along, let's say in the past 10 years? Has it been kind of a linear progression? Is it flat or is it exponential? And I know that may be a loaded question, but your impressions.
Yeah, no, it's a great question. And I think as someone also who does research that we answer to the public in many ways, I think it's a really important question. And you know, I'm coming from the camp of basic neuroscience in many respects, right? So we try to understand sort of the basic principles of the brain in order to be able to build better translational approaches. And so in the last 10 years, it might feel slow from the outsider's perspective, but if we really think about some of the amazing kind of knowledge or findings that we've had, they've been pretty incredible. So things like knowing that we have face cells, that you can take a few hundred neurons in the brain and then be able to decode the face of someone else. So an animal looking at a picture of a face, we can decode that information and essentially reconstruct it. That's a relatively new finding from Doris Tao's group, so quite amazing work. Or the notion that we have spatial maps in the brain, the so-called grid cells, or these GPS cells in the brain, which were a Nobel Prize in 2014. I think that's a pretty incredible knowledge fact that we've had. But I would say a lot of neuroscience research has really been pushing the tech to essentially, right, kind of mirroring what Hartmuth said, like a lot of the things that will really push the field forward now and has been is really the technology. And in that front, there's many, many really exciting things that we've been able to do. The ability to clear a whole brain and look at all the neurons in one chunk of tissue is incredible. That was really pioneered by Carl Dijsselroth's lab and many others now, like Ali Ertuğrul, who can clear a whole mouse or a whole human organ and try to look at the connections in the brain. These are pretty incredible findings, I would say. But I could go on about this forever, so I will yield my time, as one might say, to see what else.
One quick follow-up question. You started by saying to the lay public, it may not seem that things have evolved, but to the scientific community, it sounds like there's big jumps. Why the gap? Or why do you suspect there's a gap in knowledge in the lay, or even the somewhat technical sort of audiences? Why wouldn't they see that leap?
Yeah, that's a great question. I mean, I think the bar of information that, say, makes it into the textbook of like basic neuroscience is quite high, and for a really good reason, right? Like we test hypotheses, we don't prove hypotheses to be true. We say things that aren't true in science, right? That's sort of the scientific method. And so for something to be the level of a really tried and true fact or knowledge is quite a high bar. So I think it might look slow from the outset because there's kind of what meets this bar might look very slow, like things like grid cells or GPS cells and some of the day-to-day grindings. I mean, there are amazing, publications coming out every day in neuroscience that change the way that I and my colleagues or even the public might think about the brain. but they're not all making the cover of the New York Times, right? And so I think there's also kind of a disconnect, at least from my perspective, about how much scientists are willing to engage with the public, and that is a problem, right? Like it's scary to come on as an expert and talk to the public about these things. And I don't know how many bench scientists you've met, but you know, we're not necessarily the most charismatic group of people. We weren't hired for our public speaking skills.
With the exception of these three, I'm pretty sure that you may be right.
Yes, of course. Hartmuth and Paul, I'm sure, are fantastic. So, yeah, but I think there might be a disconnect in that way. But I think there's, with the age of the internet and all these other things that are coming, of course, that are actually not that old if you think about it, the information is available. It's about really making it accessible and understandable. And that's our job as scientists, to do that better.
If I could just add to that, Dan.
Yeah, please.
To reinforce what Mackenzie was saying, in the last 30 years, we've probably learned more about the brain than in all of history up to that point. But for those that aren't in the space, you're looking at what you saw last month to next month. The pace at which you're seeing something is relatively small, but those who are in research, for better or worse, are forced to relive the past, to look at old research, to read all the old papers, to stand on the shoulders of those that came before us so that we can make a step forward. So we have a better, I think, understanding of where what we're doing sits within the history of the art, whereas the general public, they wouldn't be forced to do such things, right? They're really just, what they're catching on social media or in the news, which are really just time snippets of what things are like today without the comparison to the way things might have been 50 years ago or 100 years ago.
Very, very good context. Well, now that you've brought that up, I want to turn the question. Before I do, actually, I just want to remind the audience, if you're listening in, you have a chance to have your say as well. So you'll see there's a Q&A feature. Alexandra had alluded to this earlier. You can go in and insert your questions. We're going to, in just a little bit, have an interim Q&A because we don't want to wait just to the end. We want to make sure you do have a chance to have your say because this is such an awesome panel. But I got a whole bunch of other questions for them. So take your time, put those in. Paul Hartmuth, question for both of you. I'll start with Paul. We talked about the brain and how far it's come. How about imaging? How far has imaging come over the past 10 years? Has it been stepwise or has it been exponential? What's your thoughts on that? Paul, start with you, then Hartmuth, yeah. You can take the second swipe.
Well, to give context again for that, it was 125 years ago. So only just, right, a couple of generations when the first X-ray was taken, right? Willem Röntgen's wife, there's a picture of her hand with a ring in it. And now, I mean, there's probably not a person in your audience who hasn't had at least an X-ray, if not a CT or MR or ultrasound, right? So it's gone from being completely new as an idea to being ubiquitous in the practice of medicine, let alone clinical trials or the kinds of advanced research that Mackenzie's doing. Mm-hmm. So again, one of the rate limiting steps had always been computing power. Well, when you look at Moore's law and the rate at which the increase in computing powers have moved forward, every single time that happens, there's an impact to imaging as well. The things that couldn't have been done even 15 years ago based on the speed or the size of certain chips, or Hartmuth could speak a bit more eloquently about PET and some of the advances made there, myself probably MR. You're getting much more rapid sequences, much finer resolution, much shorter times for patients and scanners. So you're acquiring much more information. When you look at things that are dynamic, like functional imaging, some of the things that Mackenzie's doing, the time signatures, there's so much data where that might have been untenable even 15 years ago, even 10 years ago. And now with the computing power that we have at our fingertips that you can just use on your laptop or your phone, it's remarkable. So that's all impacting the pace and the kinds of information that we can now interrogate with imaging modalities.
One quick follow-up before I turn to Hartmuth. Does this enable everyday sort of citizen scientists to have access to some of these technologies that might have been not attainable a few years ago? Could you have a garage sort of imaging ability to use imaging to, in the backyard or garage, the Steve Jobs, in the, building something that
you're getting there. You're getting there. I'd say some of the technologies, PET, because it's radiation based, MRI, because of the typical superconducting that is involved in most scanners, they're a little less approachable, but you can get an ultrasound that you could wear around your neck these days. And I'd say 15 years ago, that was maybe a stretch, right? So you're starting to approach that. I have friends that have developed a standalone brain MR scanner that you can roll into a room and scan someone's brain and roll it out. That was just approved by the FDA, I think this year or last. So it's really a question of accessibility. It's becoming more available publicly. I think in terms of the garage scientists, there are some things that can be done, but we're still a little far away in that respect.
Maybe we'll keep it out of the garage. We did hear in a previous installment of Existential Medicines around CRISPR that you have some sort of biohackers.
The biohackers, yeah.
Yeah, that are, so I wondered if that was perhaps there's an overlap with imaging, maybe not quite the same.
When you look at optical, there are opportunities. When you look at ultrasound, there are opportunities. Some of the other modalities are still a little bit out of reach, but probably for good reason in some cases.
Hartmuth, your thoughts on that, and then I have a follow-up for you more specifically about understanding the brain and function dysfunction, but your thoughts about where things have been going with imaging.
I think we've made tremendous progress in the last 10 years, especially, and I focus mostly on molecular imaging, so basically imaging molecular processes in the brain or elsewhere in the body. And when I look back 10 years ago, amyloid imaging was just starting, right? And there was no tau imaging. And now we have FDA-approved amyloid imaging and tau imaging probes. And the National Institute of Aging and the Alzheimer's Association have both developed a research framework that is based on these biomarkers measurements, right? On amyloid, tau, and neurodegeneration for classifying and staging Alzheimer's disease. Right? So this was unthinkable 10 years ago, right? And all this happened in 10 years. And the pharmaceutical companies are using this approach that was developed by Mayo Clinic and Alzheimer's Association and so forth for running their clinical trials. Right? For example, our clinical trials in Alzheimer's disease, they are driven by these biomarker measurements. Right? So we are doing amyloid imaging, we're doing tau imaging, we're doing MRI to measure neurodegeneration, and we do that to diagnose patients and to stage them and to track progressions. So we can see how much new tau, which is neuronal tangles, a patient develops in a year. Right, and so we know it's about 5% in Alzheimer's, or 6% even Alzheimer's disease. Right, so we can track that, and then we can see whether the therapeutic intervention changes that track.
And just to interject, Dan, on Hartmuth's comment, I mean, we're talking recently within some of those early Alzheimer's trials, prior to the ubiquity and availability of amyloid tracers. Patients were enrolled in those clinical trials based on symptoms, so clinical symptoms diagnosed as likely Alzheimer's disease. Come to find out, based on having the ability to use amyloid imaging to then actually interrogate and determine which of those patients have, in fact, the plaques that they are testing against. Anecdotally, some of the large clinical trials, 40 to 50% of those patients did not have amyloid in their brains. Therefore, by definition, did not have Alzheimer's disease. So Hartmuth is absolutely correct in terms of, in the past 10 years, you've gone for a case where you have a clinical trial where maybe 50 to 60% of the patients may not actually have the pathology that you've developed your drug to test against, to making sure that every single person in that trial does in fact have the pathology, so you can actually test the efficacy of that drug against that mechanism and then determine if it aids in clinical symptomatic treatment.
So just a quick follow-up for both of you then is, it sounds like that this is actually improving the quality of the trials, perhaps reducing the failure rates. Are you seeing some of the I guess the fruits of the improved imaging and the trial outcomes?
I think so, and I'm not yet, because we are just running the trial, so you can't speak about that. But the Biogen aducanumab trial is looking much more promising now, and it was also based on imaging for anti-amyloid therapy. And then Lilly basically did a huge imaging study using the expedition-free anti-amyloid sonelokimab trials, and where they learned a lot about progression on tau imaging, but which they are now, and we are applying for our future trials. Right? So I think when people say there's no therapy for Alzheimer's disease, that is true today, right? But I think in 5 years, the picture will look different, right? And that will be true because imaging will have enabled those clinical trials. That's my belief.
You'll have definitive information, I think, is
Yeah.
the key factor here. You're going to remove some of the noise and really be able to make determinations, two things. Did the drug work as intended? That is to say, if it's an anti-amyloid agent, did it remove amyloid or stabilize it? If it's an anti-tau agent, did it remove tau or stabilize it? And then, did you reduce the clinical symptoms? Did you stop the progression of the disease or the symptoms of that disease based on testing that? With imaging and only imaging, you can definitively test those first hypotheses. Did you remove amyloid or stabilize it? Did you remove tau or stabilize it? Then you have to see whether or not doing those things, making that intervention, actually has an impact on the progression of the symptoms in a patient or recovers faculty.
Right.
Yeah, and it's also been shown that, for example, tau imaging is linked to clinical symptoms of cognitive function, for example, right? And so there's a clear link between what we measure in the clinic, the clinical scores, and what we see through imaging, right? And there's a correlation there, and that's proud. Because it makes me proud because my team and I developed the tau tracer that's now FDA approved. So that's why I'm very proud of that.
That's definitely a real claim to fame. Well, I now have a quick digression for you. And just again, signal to the participants who are listening in just a few moments, it's going to be your turn to ask a couple of questions. But before we do, I've got one more quick exercise for our panelists. So before we convened, I asked them to reflect on something that they might have heard, a quote per se, that they think encapsulates the way they think about brain health, neurodegenerative diseases, and kind of the state of imaging. And some very interesting quotes. I'm going to share a couple of the quotes. And I'm going to start with first Mackenzie. Mackenzie, you had chosen a very pithy one. Certainty is the enemy of knowledge. And that was from Patricia Churchland. And from the origins of moral intuition. Why did you pick that when thinking about what this means or what it says about this particular topic?
Yeah, I picked it because I think it actually, interestingly, piggybacks really nicely on what the discussion we just had, where Paul and Hartmuth were bringing up, right? Is that sometimes we can be very certain and you need to be able to change your opinion or how you approach a problem based in the light of new information. So for example, with the 40 to 50%, I think you said, Paul, with the plaques not actually being present, you could also see that as an opportunity to go back and reanalyze that old data to say, well, the people that actually didn't have tau, how did they react to the drugs, right? And then so it might actually fundamentally change the way that you're thinking about it. And you might have been really certain that this was the right population to test on at the time, but that can change. So I think it's, I kind of think it as a quote to remain humble as a scientist and always, take in new information and think about it. So it was pithy, but at the same time hopeful in my own ways.
Well, I think that an interesting sort of comparison is Hartmuth. You had a very interesting quote from somebody you know personally. When I first saw it, I thought, what an interesting person to cite, Michael Phelps, but it's not the swimmer Michael Phelps, but just as interesting. And the quote was, there's a natural curve in life. You go up, plateau, and go down. And the only way to deal with this is by starting new curves. And by this, to remain in a state of becoming. That was a very inspirational quote. I love that. Why did you pick that? And you mentioned that you'd worked with Michael at UCLA and Siemens for a number of years. Tell me a little bit about that, why you picked that, and what that suggests for the way you view this field and how it is becoming.
Yeah, I have a lot of admiration for Mike Phelps, not the swimmer, but Mike Phelps is the one of the fathers of PET imaging, right? And so he and a few others developed PET imaging to what it is. And Mike is an incredibly good teacher, right? So he has a head full of these quotes, right? And he uses them to motivate people. And this is one of them, right? So I've heard that and many others from him. And it is true, because I mean, in life, once you stop coming up with new ideas and new innovation, it all ends, right? Because you plateau, and then you go down, right? And that's the natural things. So in order to keep going in life, you need to always come up with new things, right? And that's true in my life, my career development, but also my research projects, right? So I don't rest on my laurels, but I mentioned the tau PET agent that was just approved, but that was years ago. So we have already made probably a half dozen new agents that are in various stages of development just to keep it going, right? Because otherwise it will just end.
So it sounds like, not to put words in your mouth, that the field, neurodegenerative diseases and imaging it's not at a state of finality, far from it. It is always becoming. Very, very interesting, very fitting. And certainly not to be outdone, Paul, your quote, one that I'm a fan of, you had a couple of quotes. I took one of them: give me 6 hours to chop down a tree and I'll spend the first 4 sharpening the axe. And it's debatably attributed to Abraham Lincoln, but tell me a little bit more about that. It's a slightly different meaning than the other two.
Yeah, and I think you'll see this thematically from some of the comments Mackenzie and Hartmuth have both made. It's really about being prepared, having the right tools. We're asking very big questions. I think the people that we all work with, Dan, yourself included in your career, I know Mackenzie, Hartmuth, myself, we're asking some pretty big questions. We're talking about, what can we do to halt neurodegenerative disease in humans, right? That's a big question, and no one of us is going to answer that question. But too many times, in the pursuit of science or in the pursuit of a new medicine, people are asking the wrong questions, or they're, if every problem looks like a nail, or if your only tool is a hammer, every problem looks like a nail, right? So it's a question of making sure that you're prepared, that you're asking the right questions, so that you won't have to go back and recarve data and say, well, what if we did it this way? What if we did it this way? Then you're hunting and picking for answers. Here's a question where you come up with a very sharp hypothesis. You come up with the right tool. In this case, we're talking about imaging being the tool. You test that hypothesis. If it makes it past there, great. If it doesn't, great. I've always been agnostic about the success or failure of a drug on which I work, because my job is to make sure that the question is answered accurately. If the answer is no, that's fine. If the answer is yes, that's also fine. It just means that we've been able to, with some definitive answer, test that hypothesis. And you do see a lot of the failures, I think, in phase 2 and phase 3 trials, which are still very, very high numbers. I mean, 1 in 10,000 drugs that goes to first in human makes it to market. 95% of drugs that make it to phase 1 fail before they make it to phase 2. Something like 80% in phase 2 fail, and something about 50% in phase 3 fail. So these are huge numbers given the billions of dollars of investment. So if there's anything that can be done to say, we've set a certain threshold, we've sharpened our axe, so we know that whatever makes it past this stage has a high confidence that it's a likelihood of being successful. So I say front load imaging. Hartmuth talked earlier about the cost. Front load imaging, make good decisions, and advance candidates that have the highest probability of success.
Interesting. Great. Well, speaking of questions, now's the time. I'm going to invite Alexandra. Since I've been controlling the board and asking the questions, I'm now going to invite you to share a couple of questions from folks who have been contributing to the Q&A. So Alexandra, what is the first question that we can serve up to the panel?
Sure. So a great question here is, how does the media cycle of science hype contribute or detract from the public trust in research? Has there been a notable change with the sped up news cycle?
I'll direct that to anyone on the panel who would like to take it. Mackenzie, we kind of touched on this earlier with you. If you don't mind, maybe I'll ask you to take the first stab at that question.
Sure. Yeah, no, it's a fantastic question. And I think it plays into a lot of concerns we have with short attention span and short media cycles in general. And it also brings up a really good point of how I was discussing kind of the layman's maybe view of how science is progressing versus the researchers' kind of position of progressing, and the good points that Paul made along those lines. And so, the worry with science hype is exactly that, someone says a drug is coming to market and this is going to be amazing, and you see so much investment and billions of dollars thrown into it. But it's exactly as Paul just said. I mean, the numbers of diminishing drugs that actually make it through that pipeline is a concern. And so I think false hope is something, especially in the space of working in neurodegeneration. I mean, these are terrible diseases with very little to no cures, right? And so that is a real concern. And so I think if things are overhyped, then it does fall on the shoulders of basic research, right? Like, it's like, well, where did the science fail? Are mice terrible models? Or is the pet tracer terrible? Are we picking the wrong people? Why are these drugs? No, exactly. Yeah. Why are these things failing, right? And so understanding that actually failure is part of the process of science, I think would be much better to put in the news cycle than just showing all the flashy results all the time. So.
Paul, Hartmuth, any builds or a disagreement even that you'd like to share?
Should I go first, Paul? Yeah, yeah, at Janssen, we are quite insulated from that, right? Because we have a plan, right? And a news cycle doesn't impact us much at all, because our research focuses on treating important diseases like in cancer or Alzheimer's disease and depression and so forth. And that plan is not just driven by news cycles; it's driven by the long-term needs that we as humanity have, right? And then the budget is based on that, and that is, so we are in a lucky position where the news cycle doesn't impact us. At least my work is not impacted by it at all.
Oh, of course. I'm not saying that. I mean, my work directly isn't impacted either. But I guess the question was, how does it erode public trust in science given the short news cycle? So I think inadvertently it affects us all. If people don't trust scientists, that's a huge problem. I mean, maybe we shouldn't talk about politics, but we all know
I think, well, no, it's a fair question. And this year aside, perhaps, I think that some of the exciting bits, and again, you could also turn the public, attention the wrong way very quickly. But how many people were really aware of what clinical trials were before this year? Really? Right? Mm-hmm. Now there are people who didn't know anything about clinical trials could tell you the 3 or 4 top COVID trials that are ongoing, what vaccines are the furthest ahead, the company names of those developing them. There are people who've never considered enrolling in a clinical trial before reaching out and asking about that information. I think that's all incredibly positive. Clinical trial process and the ability to find people who participate in those clinical trials is hugely important to the advancement of science on what we're all, on which we're all working. So I think that, yeah, it can backslide on you, Dan. I completely agree. And there are times that it has and it will. But I think that getting information out to front of mind if people are available, that this is a potential course of treatment, that clinical research, what you're contributing to, what you're signing on to, I think that could help us all along the process.
Alexandra, I'm gonna invite you to grab another question, perhaps related to some of the imaging discussion that we've been having.
Yes, so this one is, how do you see new imaging technologies impacting new solutions for autism, which may start from the brain?
Maybe I'll throw that up as a toss-up. Is anybody doing work in even adjacent or direct with this that they want to comment?
My team is not, right? So I'm not the best person to comment on it. And I mean, in general, we have developed tracers that can image various events in the brain, like inflammation and synaptic density, right, and pathology mentions, and we can do board imaging to look at what parts of the brain are activated and so forth. But we haven't done, at least my team has not done any systematic studies in that to show how this is linked to autism.
Mm-hmm.
Paul,
yeah, just to say one thing of which I'm aware, and it was published a few years ago at least, had to do with an understanding that some of the anatomical morphologic changes are occurring in utero. I think that's an important question to understand, because there's a lot of discussion about when autism starts. Are you born with it, right? What are the things that factor into the progression or the diagnosis? And seeing that some of these people were able to be diagnosed in utero or see changes in utero that, by MRI in this case, that then correlated with having autism later on in life, 2 or 3 years old when they were diagnosed, or 5, 6 years old when they're diagnosed. I think that's remarkably valuable information, because then you have to know when you have to start, right? If this is a disease, or I'm not, calling it a disease, I think is a bit strong. Yeah. If these are changes that occur in utero, then you have to think about what life is like from day one and what could be done there, versus something that is acquired, or that you have, let's say with an Alzheimer's disease, a lifelong genetic predisposition, perhaps with some epidemiologic factors that weigh into that. You could intervene later in life. With autism, maybe you have to intervene very early. And so I think that gives people direction for what they need to target, as well as start to do the genetic signatures, right? That's how tau and amyloid and some of these other factors are treated, is understanding what genes drive those differences early on. If you can start to find those genes, then you have potential to find targets that can be treated with drugs that could be administered and then have the opportunity to affect that over long periods of time. So I think, personally speaking, I don't know that I'm seeing enough advances there quickly enough, and maybe consider me lay in that space, but happy to see that people are investigating and asking those questions.
I'm going to ask for one more quick question, Alexandra, and then I've got a few more of my own. And then we will take more questions from the audience at the end. But Alexandra, last one from the panel or from the Q&A.
Sure, we have a few more in here, and this last one can be about imaging's role in trial design. So the question is, are there opportunities for these techniques to enhance trial design, specifically around diversity of trial participants, and can it be used to mitigate disparities or lack of access?
Interesting. Paul, do you want to jump on that one? And then we can put that up as a toss-up.
Absolutely, yes. Absolutely, yes. Trial enrichment, and I'm speaking, in many different ways, in terms of disease progression, in terms of age, I think in terms of different ethnicities, I think it's hugely important to have that balanced and approached in the trial. So having imaging, I guess the good thing is if you're in clinical trials, access to imaging isn't a cost that goes to the patient, right? These people enrolled in clinical trials, and therefore the standard of care and their treatment is covered by the clinical trials. And I think it's important, again, another example is COVID. They're taking a lot of care and attention to make sure different populations that appear to be disproportionately affected by some of the harsher side effects of the disease or the disease itself are included in these trials. So the access to the imaging has to do with where you can recruit these patients, and then the cost of those is covered by the trial. So I think importantly, you should get hopefully more equitable data at the result of the trial. Now, access to medicines post that is a different question, but I think that the trials are being designed in such a way that it's sensitive to that, because it's the right scientific question to ask, frankly speaking.
Yeah, and I can comment on that as well, because we at Janssen, we are clearly focusing on having diversity in our trial population, right? Because we cannot have just one subpopulation. So that's why that's one key focus that we have. And then for the type of imaging that we do, PET imaging, we are largely dependent on the manufacturing of these agents, right? So we have centers around these manufacturing sites, which oftentimes are only like maybe a dozen around the world, right? So and that's where we recruit people to. But diversity is clearly an important aspect. And that's also true in my role as at the foundation of the National Institute of Health, where we are looking at research programs, clinical research programs, as well as discovery programs, where diversity is also a key focus. Right? So that's what we really have an eye on.
Great.
Maybe
all right.
Well, can I actually
yes, please.
Yes. Maybe and be a little bit provocative? I thought that's fine. So I'm just curious, especially at the NIH and your experience, Hartmuth. I mean, having diversity is also a matter of access and people knowing about clinical trials, right? Paul pointed out a good point that like maybe many people didn't even know about clinical trials now, but I mean, I really see a need for having a much better centralized database of things that are searchable by the public. I mean, even searching clinical trials from a researcher perspective with kind of that privileged position I have is actually not straightforward or trivial to find all the current clinical trials and testing drugs. So what can we do collectively together to make this more accessible actually from a researcher's perspective? I'd be really interested in your thoughts on how to do this better.
I think it's actually very accessible, right? Because I mean, you just go to all clinical trials are registered on clinicaltrials.gov, right? And so if you have a condition, you just go there and type it in, and you get a whole bunch of trials, and also the sites that are recruiting, and what this trial is about, and what therapy is being involved. So as long as you have a computer and know how to open up an internet browser, you should be able to get to that information.
Yeah, I might push back though, because clinicaltrial.gov is not the most straight. It's not like Google search, right?
No,
like, it's not straightforward. You need to use the right terms. And so I think it's actually, you're talking about populations, maybe they don't even have computers at home all the time. And so this is not, I mean, this is a much broader thing. Like that's kind of what I said, coming from a privileged position, like PubMed, clinical trials, great. But I would push back that it's easy. So I would encourage those that are in the position of making drugs and have the money to make public resources available. That would be a fantastic thing to invest in. So.
And this is happening by the patient advocacy groups also, right? And for Alzheimer's disease, you have the Alzheimer's Association, and they're really focusing on that, right? And then for Parkinson's disease, the Michael J. Fox Foundation has done an amazing job in doing that service, right? Then in breast cancer, there's patient advocacy groups, and you get emails from them with new trials all the time if you have breast cancer, right? So I think there's a lot has happened in the last few years to make the public aware of what's going on.
Yeah, no, that's encouraging. Yeah, thanks.
I'm gonna turn with another question. This is going to come back to you, Mackenzie, a slightly different topic. We started talking earlier about the work you're doing with mice, and I'm interested to know or to hear your thoughts about how does the work that you're doing, how might it potentially influence the work that Hartmuth or Paul or other folks are doing in humans with neurodegenerative diseases? How has it, or how might it actually influence that? And how does imaging play into that?
Yeah, great question, of course. Yeah, so I mean, of course, mice are not a replacement. They're not tiny humans. They're not a replacement for it. We're really looking at a different level, right? Like we're really trying to understand cellular interactions and over longer timescales. So many of the imaging modalities, like you do a PET scan, but that might be a snapshot in time, and then a patient comes back in several months, where we continue, potentially have access every day. And also can monitor behavior and the outcomes of interventions continually, right? So part of our work is actually to build artificial intelligence-based tools to monitor behavior and not just look at, say, these, chunked time points of, say, a mouse lived longer because it got drug X, but actually did they have a change in quality of life with drug X? I mean, that is something that's really hard to do in people and something much more accessible in mice. And especially in the last 5 years with the advent of deep learning, I mean, the toolbox has literally been broken open, right? And so we're really in, I think, unique position with mice to try to help in this ways. Does that mean that all the, drugs and things that we test in mice will actually work in humans? Absolutely not. But I think once we try to understand more of the basic neuroscience of how these drugs actually work on neural circuits, then we're getting into a position where we can start translating some of these findings, hopefully into Paul and Hartmuth's hands.
I'm curious, one quick follow-up for you is, any particular insight that you gleaned working with mice where it applied, or you were able to apply it cross species? Any interesting examples?
Yeah, I mean, I think many interesting examples. One that I've already touched on, just to keep continuous, the notion of grid cells was discovered first in mice and rats, so in rodents, and that's been shown in humans and non-human primates, essentially all these different scales. Bats, animals that are very different than us have maps of 3D space. And this is sort of a universal thing that was first tested in animals because it was much easier to, say, move a physical world. I mean, I'm sure many of you have seen scanners. They're not the most mobile things to carry around on your head, but animals can do that, right? Because we make these very miniature little devices, and it's very, in that sense, non-invasive or minimally invasive to be able to do these types of things. So I think that's one really beautiful knowledge example that we've been able to glean from mice to men, if you will.
And that's a good reference there. I'm curious, since you brought up about like miniature scanners that might be on a mice's head, it makes me think of what new technologies or what's on the frontier. And I guess I'll put this out to all three of you. What's on the immediate frontier, the next probably 2 to 5 years, that you think can have an impact, not just on neurodegenerative disease, but from an imaging standpoint? Do you see big changes, or do you see sort of breakthroughs that could have a broader impact? And so I'll throw that up open for anyone to grab as a question.
I'll say, and Mackenzie already said it, it's machine learning.
Mm-hmm.
So it's not necessarily an imaging advance, but there's, getting away from the traditional endpoints and the traditional timeline, as Mackenzie said. You can find signals in data by using things like machine learning that you can't get by looking at one static image as a trained radiologist at one point in time. And you can look at things that might be missed by the human eye because you're looking again at a static image at one point in time, or even in some cases at PET, a dynamic image, and it's hard to interpret some of the changes in signal that you're seeing because they're ocular resolution, but they're not for a machine. So there's a lot of activity in that space that I expect, if it doesn't just make the practice of medicine when it comes to radiology or research in this space vastly better, it's going to make it better, right? So it's it's applications of this. I'm not talking about moonshot. I'm not talking about replacing radiologists. I'm talking about augmenting our capability to find signals and see information that are otherwise difficult to see. Here's cases where you've got machines that can, better diagnose chest X-rays. Working on ones that auto-identify oncologic lesions. I know friends that are, better segmentations of the brain based on our knowledge, as opposed to some of the more anatomically defined. I mean, someone just said this is this area and this is this area, but practically speaking, is that true? Now you have machines that can work in the background that better define that based on activity, based on the connectivity of the brain. I mean, the human brain is potentially the most complex structure on the face of the earth in terms of its connectivity. So to think that, we'll take a snapshot in time and answer all the questions is a challenge. But aided by some of the advances in machine learning, I think we're going to learn a lot more information about some of the data we already have in hand.
And Hartmuth, we had chatted ahead of time a little bit about it's not just imaging that you see as being the big boost or the advancement of technology. There are other things in combination that complement imaging that you had suggested may be game changing. Can you comment a little bit about kind of what are some of those things, or what in particular do you think might be accelerating development alongside imaging in the future?
There's three things, right? So I agree with everything that Paul and Mackenzie said earlier regarding machine learning, and so that's a given and very important to happen. But with respect to other aspects, I think there's three things. One is we need new sources of information, so new imaging tracers that give you new information that you currently don't have. And for example, for Parkinson's disease, we need a tracer that measures alpha-synuclein, which is part of the key pathological hallmarks of Parkinson's disease. Or we need, but that's one thing. Then the second thing we need is easier access to these technologies, right? I mean, we touched on that earlier. How do you get to an imaging scan, right? And that is best done through a blood test, right? So basically, you do a blood test, you see, okay, you have a higher risk for something, and then you get an imaging test, right? And so we need better, simpler, non-invasive tests. And then the third thing is reimbursement for these tests, right? Because nothing will happen unless there's some kind of financial coverage for these blood tests or imaging tests. And that drives innovation, right? Because no one's going to innovate if there's no, at least you don't break even, at least, right? And so these are the three things that I think will have to happen.
So it's not just the technology, as you brought up, it's about access is a key part. And we've heard that now, not just now, but before as well, a couple of times, that access and economics can actually drive the proliferation of the technology. One other question, and I'll just signal that we will take more questions from the participants. So we'll stick on for a little longer with Q&A. But I've got a couple more questions for our panel, and then I'll be a chance for you to have your say again. So please feel free to input some of your questions into chat. Paul, Mackenzie, Hartmuth, a question about patient care. We haven't gone deep in talking about what does this mean for patients? What does it mean for providers or physicians, as it were? How is this changing the way kind of patients are making decisions? How is it changing the way, or how might it change the way physicians are behaving in clinical practice with the advancement of some of the kind of the movement towards imaging as a mainstay? Your thoughts about that question?
Just to start, I think it is aiding in physicians. I think they're becoming more accustomed to what the information is available in these spaces and understanding the limitations of a clinical diagnosis. So they're using it for differential diagnosis. Do you have Alzheimer's disease or frontotemporal lobe dementia? These are questions that they probably couldn't have answered 5 years ago, 8 years ago. I think a positive amyloid or tau test will tell you that. A tau is also in Hartmuth, again, thanks for your contribution in this space, looking at traumatic brain injury. Tau is associated with traumatic brain injury, which we understand is also an important issue across the world. So there's different advances that physicians now have at their fingertips that let them better treat their patients, better understand the epidemiology of the disease or the pathology, and therefore have a better opportunity to treat it effectively. I think it's important. I will also say, though, in terms of access, I would be, and I don't think I'm out of turn to say Mackenzie and Hartmuth would probably agree, everyone would be happy to have a spit test that diagnose Alzheimer's disease, right? We would love to get to that part where it's an accessible biomarker and the cost is dirt cheap. We're not advancing imaging for the sake of, I don't own an imaging company, right? The point of what we do and what I do is to ask the question, and imaging is the only way to answer that question. Once we've answered that question, if the medicine's approved and you can get diagnosed based on, a sweat test or a spit test or a blood test, that would be amazing. It's being certain that that test is associated with the pathology and that drug can work against it. So I think imaging enables the access to medicine because you've answered the hard questions through research all the way up through that, phase 3 clinical development and approval, and post-approval, maybe the hope is that you wouldn't necessarily have to use imaging.
I 100% agree with everything Paul just said, right? And I see imaging largely as a research tool, actually, right? A clinical research tool, as well as a preclinical research tool on mice, rats, and so forth. And once that is done, and we've done better research and developed drugs, then I think we can move on to non-imaging ways to diagnose patients. That's the way I see it.
I have I'm sorry, go ahead, Mackenzie, please.
Oh, no, I was just saying I fully agree. I think I have nothing more to add on that.
I have one sort of a kind of wild card question. The thought of crowdsourcing. So I'm not sure how much crowdsourcing images has come to play yet. Crowdsourcing is used in all different modalities, from NASA having crowds of people helping them solve aerospace issues, to Foldit, which is a protein folding application where there are hundreds of thousands of gamers doing protein folding who have no training whatsoever, but they're given gaming instructions. I'm curious to put this question to you. How might, or how is crowdsourcing, how might we be able to tap into the power of citizen science and the crowd to be able to interpret or to look at images to help advance our understanding, even perhaps even lead to a breakthrough? So I'm going to put that kind of provocation out there to you three. Any input into that?
Yeah, maybe just a quick, before I pass it on, I think the issue with that is patients' privacy, right? And because it's very hard to make patient data available for crowdsourcing applications, right? So that's the key with holding us back. So I think it's easier if you deal with animal imaging, right? And but with human imaging, I see it almost as impossible to make it available. But that's just my take.
Paul, Mackenzie, thoughts?
I might chip in. So, a large part of the work I do is open source science and open source development and releasing these things. And a big part of that is in computer vision. And so Paul touched on this earlier about discussing machine learning, which is, there's a way that we have to train these algorithms to be better than or as good as humans, right? And the way that we do that is on data, and you need diverse data and you need a lot of it, just as part of it. So there are challenges, for example, unlike PET tumor segmentation challenges, where computer scientists can chip in and try to build better algorithms. And so it might not be citizen science in the conventional sense, but it's open science, which I think is a really positive thing. And many of the scans that, a mouse on a scan isn't like crazy different from some of the computer vision challenges that come up with human scans. So that is a really nice way to actually be able to sort of transfer the learning that we can use on, mice probably care slightly less about their privacy, or we as humans care less about their privacy, but able to translate that to human, I think, is a really, really fruitful avenue going forward in terms of crowdsource and citizen science.
Yeah, I completely agree, both with Hartmuth and Mackenzie. I think what you see is, along the lines of what Mackenzie said, is it's really technology transfer. It's not necessarily the general public, which there could be opportunities for them to contribute there. It's more along the line of, people who look at satellite images. Is that different than looking at the brain? Yes. Is it similar to looking at the brain? Also yes, right? Looking at traffic patterns. Is that similar to looking at the brain? Yes. Is it different than looking at the brain? Also yes, right? So there's different, very advanced, very incredible tools that exist in other areas that, frankly speaking, the three of us might not have come across in our training, or might not be aware of, or the people that work for us may not be aware of, that someone out there is doing the thing that we need to be done every day of their lives, is simply as they brush their teeth, and we would kill for that opportunity, right? So making the question, and again, it goes back to sharpening the act, making the question available and helping to find people who can help answer that question, I think is the crowdsourcing opportunity that you speak of.
Great. I'm going to stop here for a moment. A couple more questions from the crowd. Alexandra, a couple things that haven't been asked yet that the panel can weigh in on.
Sure. So this one is more of kind of in the news, but what are some current thoughts around Neuralink's latest announcement?
I'm actually going to direct that to Mackenzie because we started with that, and
You took the words out of my mouth.
Yeah, just before we started this publicly, there was an internal discussion on this, and I think Mackenzie's infinitely more qualified than I am to answer that.
I'm not sure, but yeah, no, I mean, of course, I think what Neuralink is doing and making one neuroscience cool for the general public, I think is really exciting for many of us. I think they're using a lot of really amazing things that have been developed in basic science, so I think that's a positive thing. I think what they want to do, I've no affiliation with Neuralink or no financial anything with them, so let's put it that way, first of all. I think what they aim to do is a huge question and a big challenge. And I think the engineering that they need to do to make it accessible to humans eventually is a really, really big goal. And so it was really encouraging to see them, like wireless recording of thousands of neurons or even hundreds of neurons was a big advance. Like the tininess of the chip is very exciting for us. Of course, this was more of a PR event. It wasn't a publication, so the proof will be in the pudding for many of the scientists out there. So I think there's sort of two camps in general. I think there's the neuroengineering side, and people are excited about the tech. And there's the science side, which of course there wasn't new knowledge announced or new things like that, but hopefully those type of advancements can come. And we all hope that they do that kind of hand in hand with basic neuroscience as well. So.
And I'll say this because I don't think Mackenzie will. It appears that Neuralink was probably using some things that Mackenzie's worked on in her career, which is pretty exciting.
Yeah, they definitely are using markerless motion tracking, which is one of the things that we've developed and made open source, and thousands of people are using, so it's quite exciting. Yeah.
And for those who are naive to Neuralink, couple of the quick snapshot. What is Neuralink? Why did this make kind of a media splash?
I don't know their tagline, but they're a company founded by with Elon Musk, and they've sort of recruited some really amazing scientists over the years from top institutes across the United States to come together to tackle the challenge of doing sort of brain-machine interfaces. I think they have even bigger aspirations, but in general, they have very good goals in order to restore human health in patients that have spinal cord injuries or other neuroprosthetic needs. So I think it's quite a big challenge and an exciting time to see people getting into that game, because it's really hard work. Like Dan, you asked at the beginning of the day, what is my day-to-day life like? And it's slow and it's painful and experiments fail. And it is a lot of really, really challenging stuff. And you need to be not only a good surgeon, you need to be a good technician, you need to be a good computer scientist. And so seeing a company take on this challenge, I think is quite exciting, to be honest. So, yeah.
Right. Alexandra, I think we have time for one more question. What's one more question that the panel would love to answer?
Sure. This is a bit of a bigger one, but I'll try to pare it down a little bit. So it's the question was written to say, it's clear that these imaging modalities can be powerful tools to rule out disease, but are there also examples of way they can redefine disease?
Yeah. I'll go back to my earlier example. Alzheimer's disease is a set of clinical symptoms, except for that it's not, right? That's how Alzheimer's disease was always approached. It come to find out through CSF sampling and particularly through imaging that many of those people, half or so, didn't in fact have Alzheimer's disease. They had frontotemporal lobe dementia or something else that mimicked the symptoms. So what you have is a better ability to understand the categorization of the disease. I think Hartmuth could speak to this. Looking at Alzheimer's disease, there's amyloid pathology, there's tau pathology, there's neuroinflammation, sometimes there's all three, sometimes there's two, right? Sometimes there's more of one than the other. The way that tau progresses through the brain more mimics the clinical symptoms than amyloid did. So is amyloid really the key, or is it tau, or is it neuroinflammation, right? These could be different diseases, or within a population that have some of those pathologies. There are people who die at 100 years old, sharp as a tack, with brains full of amyloid, well, why didn't they have Alzheimer's disease? Because there was something else missing in the pathology of the disease. So it gives us the ability to categorize those different pathologies and associate them with the symptoms, and therefore better give physicians better tools to treat those people. And hopefully, you know, what we're all working on is better medicines that can treat those separate categories. You know, test the hypothesis and then find the treatment for it.
Hartmuth,
in
last
addition
word
to
on
all
that
this,
one?
yeah, in addition to all this, which is all the case, you can also look at comorbidities, because a neurodegenerative disease is not just one thing, right? Because it's sometimes often several things that happen at the same time. And through imaging, like using the techniques that Paul just explained, you can really separate that, right? And you can find PD patients with amyloid in the brain and so forth. I mean, we've even shown that people with major depressive disorder during an active episode of depression have higher inflammation in the brain than people that are responding to treatment, right? So that shows you how imaging can actually help you understand disease, such as even depression, much better. So I think we are learning a lot through imaging these days.
Great example. Well, I hate to do it because I have a whole lot more questions to ask you guys, but I want to first thank our panel. I'm going to turn and thank you guys, and thank you, Hartmuth, and
You're
also Mackenzie. Thank you very much. You guys did awesome, really provocative. If there are questions we didn't get to, we may ask for an overtime to answer a few of the unanswered questions. But for now, for me, I look forward to our 9th installment in the future with you all. Big thanks again to our panelists. I'm going to turn it back to Alexandra to wrap things up.
Thank you. And that was a great panel. Thank you all so much for spending the last hour with us, and thanks to our guests. Please be on the lookout for a post-event follow-up from New Lab, and we'll include a recorded video of today's event, any materials that might be relevant, and a survey that helps us with all future programming. So if you're interested in joining New Lab, 2 main ways that you can get involved are through our studio programs, which are corporate or civic focused, and of course startups, you know, always look into us. But we are so happy that you joined us today. We love existential medicine, and thank you again to our panelists and to Dan.
Thank you. Thank you, Dan.
Take care, everyone.
Yeah.
Thank you, Dan.
Thanks, Paul. Yeah.